Hello, everyone. My name is Medhini, and I'm a sophomore bioengineering major at George Mason. This summer, I worked with Dr. Fernando Munt and my mentor, Dr. Juan Cerebral, on a project where we computationally modeled cerebral blood flow to optimize and evaluate treatments for ischemic stroke. So a stroke occurs when insufficient blood flow causes a localized set of brain cells to die in a human being. About 87% of stroke victims experience a type of stroke known as acute ischemic stroke, which occurs when a person's brain doesn't receive enough blood due to the presence of an occlusion or blood clot in a cerebral artery. In order to clear the clot, a catheter is often inserted into the affected artery to either vacuum or dissolve the clot. However, the catheter itself can further contribute to blockage of blood flow in some individuals. The main cerebral arteries are known as the circle of Willis and are interconnected via smaller vessels known as collaterals. Each human being exhibits a unique level of collateralization, which is important because collaterals can reintroduce blood into blocked arteries and impact overall blood flow. Generally, people with more collaterals will be impacted less by the presence of occlusions as their blood will continue to flow throughout the brain and its arteries. On the other hand, a person that lacks collaterals entirely is at much higher risk of having a fully blocked artery when they develop an occlusion, which can have much more dramatic effects. While this can be hard to examine in practice, many potential scenarios can be simulated with computational models, which is what we did this summer. In order to understand how collateralization level and catheter size impact blood flow during treatment of an ischemic stroke, we created one-dimensional computational models of the circle of Willis, with an occlusion in one of its arteries, known as the left middle cerebral artery, or LMCA. Our models measured blood flow and pressure throughout the brain across 5 relative collateralization levels: no collateralization, poor, fair, good, or excellent, and 3 catheter diameters: no catheter at all, a diameter of 0.05 centimeters, or a diameter of 0.1 centimeter. Our models were generated by programs developed in Dr. Cerebral's lab, and we referenced images of the brain as well as Circle of Willis templates from existing research. We started our data collection by generating graphs that compared blood flow in each of the main Circle of Willis arteries at the 3 catheter sizes that we tested. We generated graphs for each collateralization level, but the one seen here demonstrates flow in a situation with excellent collateralization. Here, we can see that all of the blood vessels still exhibit some flow, even though there was an occlusion in the LMCA. While flow in the LMCA is lower, it is still present because the collaterals enable blood to still flow through this artery, even with the occlusion. We also generated 3D visualizations of blood flow and pressure using a software previously developed in Dr. Cerebral's lab. The video here demonstrates a situation with poor collateralization. In this model, the occluded side, or the left side of the brain, since we're viewing it from the back, has a much higher blood pressure due to the presence of the occlusion, while the unaffected side, or the right side, is in green. By rotating the brain around, we can see how the poor collateralization doesn't enable enough blood to flow from the unaffected side of the brain to the affected side of the brain. And so we see this significant discrepancy in how the blood is able to flow due to the collateralization level. The next step in this process would be to generate 3D models of the Circle of Willis that we can then use in testing. And we can create these using the 1D models and the brain visualizations that we've produced this summer. From the data that we have gathered, we've been able to conclude that in certain cases, a lower level of collateralization and a larger catheter size can contribute to an extra decrease in blood flow through an affected area during an ischemic stroke event. Ultimately, our research will be able to help patients, especially those who are at higher risk due to lower collateralization levels, receive optimized treatment for ischemic stroke so that we're able to keep them safe while still treating their condition. I'd like to thank Dr. Lee, Dr. Cerebral, Dr. Munt, and everybody else involved in OSCAR for this amazing opportunity this summer. And thank you very much for listening.